Why do some cancer patients respond to immunotherapy while others don't? New research in Nature suggests that genetic elements left over from ancient viral infections may play a role, demonstrating that antibodies targeting the proteins encoded by these sequences may boost lung cancer patients' response to immunotherapy. Today, we will discuss these results and what they mean for cancer treatments. We will also explore the broader, deliberate application of viruses for treating cancer. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. The study we are discussing today focuses on lung cancer, the leading cause of cancer-related deaths worldwide. There have been notable advancements in cancer therapeutics in recent years, particularly in the context of immune checkpoint blockade, or ICB. This treatment method exploits the body's immune response to fight cancer. It involves inhibiting interactions between checkpoint proteins, such as PD-1 and PD-L1, on T cells and other cells, including normal or cancer cells. These interactions turn off the T cells and prevent them from destroying their binding partners. Cancer cells can exploit this system by upregulating checkpoint proteins to evade immune destruction. Blocking these protein-protein interactions prevents the off signal from being sent and allows T cells to remain active and ultimately to destroy tumor cells. The problem is that ICB is not always effective. In fact, roughly 70% of patients with lung cancer fail to respond to the therapy, though why this is unclear. However, B cells, particularly the antibodies they produce, may have something to do with it. Research suggests that areas of B-cell expansion in tissue around tumors can be used to predict whether a tumor will respond positively to checkpoint inhibition. The researchers here wanted to better understand these immunological underpinnings of the checkpoint blockade response. Using a mouse model of lung adenocarcinoma, the scientists tested the effect of ICB, specifically anti-PD-L1 therapy, on mice with cancer. They found that ICB treatment promoted survival of the mice. Moreover, compared to control animals, those treated with ICB exhibited increased titers of anti-tumor antibodies. These antibodies bind antigens expressed by tumors and, in doing so, flag cancer cells for destruction by immune cells. In this study, the scientists discovered that a dominant target of antibodies in mice with lung cancer were surface proteins associated with endogenous retroviruses, or ERVs. But what exactly are ERVs? Well, retroviruses such as HIV are viruses whose RNA genome is reverse transcribed to produce DNA when they infect a host. The DNA is then integrated into the host cell's genome. ERVs are DNA relics of retroviruses that infected our ancestors thousands or even millions of years ago. The DNA from these ancient retroviruses became stable elements of the genome, passed from generation to generation. In fact, over 8% of the human genome is composed of sequences of retroviral origin. Because these sequences have been modified by host processes and mutated over time, ERVs don't generally encode for infectious viral particles. They do, however, produce transcripts that can encode parts of a virus, for example, envelope proteins. ERVs play an important role in the regulation and expression of genes, including those involved in immune responses. Furthermore, in cancer tissues, ERVs are generally upregulated compared to healthy cells. With that in mind, the researchers here found that ERV glycoproteins were highly expressed in mouse cancer cells. Moreover, anti-ERV antibodies helped prolong survival of animals treated with ICB. The findings implicate anti-ERV antibodies as key players in anti-tumor immunity and immunotherapy responses. Dr. George Cassiotis, the senior author on the study, noted in a recent BBC News article that, essentially, ERVs trick the immune system into believing that the tumor cells are infected, and it tries to eliminate the virus, acting as sort of an alarm system. Investigations using samples from human patients with lung cancer from multiple cohorts revealed similar results. The scientists found that plasma from 45% of patients with lung adenocarcinoma that were tested, out of 80 patients total, had antibodies reactive against a glycoprotein from a human ERV. As was observed in mice, when the scientists tested titers of these antibodies in 7 patients with lung adenocarcinoma who were receiving immune checkpoint blockade, they found that treatment boosted anti-ERV antibodies in all of the patients. Furthermore, when the researchers examined RNA sequencing data from a separate cohort of patients, they found that expression levels of another human ERV were higher in patients with lung adenocarcinoma who responded to ICB compared to those who did not. The expression levels also correlated with overall patient survival. Now, though these data are correlative, collectively the findings point to involvement of ERV expression and ERV envelope targeting proteins in anti-tumor immunity and successful ICB treatment. These results are important for several reasons. For one, they advance understanding on the links between humoral immune responses, specifically anti-tumor antibodies, cancer, and immunotherapy, which the authors note could be used to inform methods for targeting the expansion of B cells to predict the response of or sensitize tumors to immune checkpoint blockade. Moreover, in a recent article in The Guardian, Dr. Cassiotis noted that additional research could lead to strategies for developing cancer treatment vaccines comprised of ERV genes to boost antibody production at a patient's cancer site and, ideally, improve immunotherapy outcomes. Essentially, we may be able to therapeutically capitalize on the genetic relics of our ancestors' viral infections. Now, this study illustrated an example whereby viral infection, specifically the integration of viral DNA into the host genome, indirectly helps combat cancer. However, viruses are also deliberately deployed to defeat cancer cells. These viruses, called oncolytic viruses, or OVs, represent a new class of cancer therapy. They come in a variety of different types, from adenoviruses to herpes viruses, and defeat cancer by directly killing or lysing tumor cells, or inducing anti-tumor immunity, similar to what was outlined in the previous paper. OVs are generally engineered to boost immune stimulation and/or remove pathogenicity-associated genes. However, administration of OV therapeutics can be challenging. Most are administered intratumorally, though intravenous delivery is most desirable given its potential for reaching primary and metastasized tumors. Still, issues like inefficient targeting of tumor sites, risk of cytokine storm, and more make this route of administration difficult. In a new report in Journal of Virology, researchers propose a solution that draws some loose parallels with the study we just discussed. That is, the scientists explored whether cDNA from an oncolytic Coxsackie virus, an RNA virus but not a retrovirus, could be deliberately integrated into the genome of human cells. The idea is that expression of the cDNA leads to the production and release of viral particles, potentially at the site of a tumor. To do this, the researchers integrated cDNA from the Coxsackie virus in a human kidney cell line and passaged the cells multiple times. They found that viral cDNA expression remained stable after passage, and the cells continued to release viral particles into the surrounding media. Moreover, there were no changes in the morphology, growth rate, or virus generation ability of the cells over time. The findings support the concept that cells can be used as carriers for the systematic delivery of OVs. To that end, more research would be needed to optimize the system, such as fine-tuning the expression and production of virus in specific environments. That is, the tumor microenvironment. In addition, these experiments were done entirely in vitro. The findings have yet to be explored for in vivo applications for combating cancer, though this is an interesting first step. So what have we learned about viruses and cancer immune responses and treatments? Well, first, antibodies targeting endogenous retroviruses play an important role in anti-tumor immune responses and may contribute to immunotherapy response in the context of lung cancer. On a broad scale, viruses are emerging as key players in the field of cancer therapy. Oncolytic viruses can and are being used to kill tumor cells and modulate immune responses. New research suggests that incorporating viral genetic material into cells could be a viable delivery method for OV therapy. Ultimately, these findings point to the importance of viruses and their integrated genetic material in cancer immunity and therapeutics. And that's all for today. Be sure to ring the bell to be notified whenever a new Microbial Minutes drops. I'm Madeline Barron. As always, thank you for listening. Thank you to Ray Ortega for production, and I'll talk to you next time.